us?
Nope.
All right. Thanks, ladies. Here's our next one. Molly and Mara, tell us a little bit about dopamine. This is probably one that a lot of people have heard of.
Okay, so we had dopamine, and the source of signal is that it's found in the central nervous system in the hippocampal dendrite gyrus and the subvascular zone, which is in your brain. And they're also found in the kidneys and vascular, which is like your blood system. And the source of the signal is the dopamine.
It is a paracrine signaling, which means it involves neurotransmitters like dopamine that carry specific signals from one cell to another. And dopamine molecules follow distinct dopamine pathways in the brain as neurotransmitters carry signals at the synapses.
And then they are a type of G protein for the type of receptor, and there are 5 different types of them: D1, D2, D3, D4, and D5, and they are all responsible for different jobs. And D1 and D5 tend to be grouped together, and then the subgroup, like the separate group of them, is D2, D3, and D4, because those groups tend to do the same things. And the summary of the pathway is the neurotransmitters are made in the brain, and then they get released when you're experiencing reward or pleasure, and then they get sent to your central nervous system and then to the different cells that have dopamine receptors on them. And then Miss Ness-Deden, can you play the video link?
Yes, does it have sound?
No, you don't need the sound. It's just like 40 seconds and it's the animation.
Okay, I just need to present it differently if it has sound in here. Hang on.
Okay, so those like blue things are the dopamine, and then they're getting attached to the dopamine receptors, which is happening right now.
Cancel. All right.
And then in the picture, you can see what the video is showing, but it's the dopamine being released from the brain, and it reaches the D2 dopamine receptor, which causes enzyme adenylate silase to be activated, and CAMP, which is the secondary messenger, is produced. Striatal neurons are also cause excitatory neurotransmitters to be released to the brain, and this is what causes the brain to expect a reward or pleasure from dopamine. And throughout the rest of the body, the pancreas produces insulin, and kidneys increase sodium excretion. And some other information is that antipsychotic drugs that can be used to treat diseases like schizophrenia block dopamine receptors. And by blocking the receptors, symptoms are less extreme. And if your body has too little amounts of dopamine, it can lead to Parkinson's disease. And too much dopamine can cause mania or hallucinations, which are experienced with schizophrenia.
All right. Thank you, ladies. And our third neurotransmitter is serotonin. Has Macy joined us yet?
Not yet. I texted her, but I didn't get a response, so.
Okay, let's skip this one. We'll come back at the end if she shows up, or if you just want to give us kind of a short version today. Would you rather do it right now, Emma, and just be done, or do you want to wait?
I can wait.
Okay, we'll come back to this one at the end. Okay, so those are the neurotransmitters, which are all, you heard the word a couple times in there, paracrine signaling, which just means that the signal is only going a short distance. And depending on the receptor, some of these slides that I show in between are in your note packet. I skipped over quite a few of the ones in your note packet that has some extra information, but this is just to kind of tie together the cell signaling. As you heard in there, some of those neurotransmitters bind to ion channels, and some of them, they mentioned some of those those G-coupled proteins that initiate signal transduction pathways and amplify the signal and can increase the chance of an action potential in the next cell. One other thing I wanted to mention about those, the same neurotransmitter can sometimes bond to different types of receptor proteins, especially if they're interacting with something like muscle, to lead to different types of effects. So this example on this slide just shows you how acetylcholine, which is one of the ones you just heard about, when it interacts with a skeletal muscle, can lead to the promotion of muscle contraction because of the protein that it bonds with. But in a heart muscle, it's the same signaling molecule, but the receptor protein is different. And so it ends up triggering a different pathway that leads to a different response. In the heart muscle, it actually inhibits or prevents muscle contraction. It's the same signaling molecule, but different receptor proteins change shape in different ways and lead to different effects. And you'll see that again when we get to hormones, that the same signaling molecule can lead to different effects depending on the molecules that make up the rest of that pathway. So that's one thing to notice as we go through here today. Real briefly, I am just going to talk about the immune system. Nobody in this class actually chose these topics, but cell signaling in the immune system is important as well, and happens a little bit differently. This slide I added today because nobody did this in this class. And so what you're looking at here is just a super simplified summary of how cell communication works in the immune system. And the important thing here is to notice that the cells need to be in direct contact. When a white blood cell ingests something foreign, a virus, a bacteria, something in your blood, it breaks it up and it actually sticks a little piece of it out the cell membrane. Those are those things on the left that look like little pencils coming out of that white blood cell. But then you can see the helper T cell actually physically binds with it. They actually touch each other and connect. And that signals a conformational change in that protein in the helper T cell that triggers that cell. It triggers a signal transduction pathway in that cell that leads to a couple different effects. It releases chemicals that activate cells that actually go out and kill other infected body cells. That's what you see on the top right. It also activates what are called B cells. That's another part of your immune system. And B cells are the ones that produce antibodies. So you might remember we looked at this diagram when we did the ELISA lab when we were still hybrid. And this kind of shows that whole flow. The part on the left is called the humoral response. That's the part that produces antibodies and defends kind of against the pathogens in the blood. The one on the right kills infected cells, but all of the immune system, the type of signaling happens when the cells are actually in direct contact with each other. So instead of a molecule that just kind of diffuses through, the two cells actually are touching parts of their cell membrane, and that's what triggers the effect. Plants also use cell signaling pathways in their immune responses because plants defend against viruses too. And I just left one slide in here to kind of show you that a molecule from a pathogen, like maybe the tail of a bacteria, the flagella coming off a bacterial cell, can trigger a signal transduction pathway in a plant cell that can lead to that cell producing antimicrobial chemicals to kind of fight against those invaders. Or to like toughen up the cell wall and make it hard for other pathogens. So there are different types of defense mechanisms that can happen, but they use that same type of signaling. So immune signaling has more to do with direct contact. The other system that has a lot of signaling molecules is the endocrine system. So the endocrine system is a little different than the nervous system because the nervous system goes really quickly. And those signals only go across those short synaptic spaces. The endocrine system, those hormones can travel much, much farther. And since they circulate through your bloodstream, the signal lasts a lot longer. And so you can have those hormonal effects that can last for hours or days, depending on exactly what type of hormone it is. So the next several groups are going to teach us about some hormones and the effects of those. So we have 4 of these in this group. And so the next one is going to be, we're going to come back to those. I rearranged the order at the last minute. We're going to talk about aldosterone. So Hannah and Abby, take it away.
So I'm going to do like the source of signal. So aldosterone is a steroid hormone, and then it's produced in the cortex of the adrenal gland. And then the types of signaling it has is it's released by the adrenal glands, which are a part of the endocrine system, like you already said. And then it's stimulated by an increase in potassium.
And the aldosterone receptor acts as a ligand-dependent transcription factor, and the cellular response is that aldosterone activates sodium and potassium channels or pumps, leading to sodium reabsorption and potassium secretion.
And then the other info we have is that it regulates the salt and water balance of the body, and it has an effect on the blood pressure. And then we have a video. It does have sound, so I don't know.
Yeah, let me try. I just need to present a little differently. So give me 1 second. I have to present a tab instead of the whole screen. Give me 1 second. Oh, go away. And I think I can just continue doing this. Oh, hang on. I need to click on the video first and then do it. Hang on. Sorry, this was trickier than I expected. Let me try this and see if it works. If I make that full screen, can you see this? Let's see how this shows up. Yes. All right. You want it from the beginning?
I made it so it's like timed for the part of the YouTube video, so
Okay.
I could just start.
Okay. Oh.
As aldosterone is hydrophobic, it diffuses into the principal epithelial cells of the late distal tubules and cortical collecting tubules, and in the cytoplasm of the cell will bind with the mineralocorticoid receptor to form an aldosterone receptor complex. This complex then diffuses into the nucleus of the cell. Once inside the nucleus of the cell, it upregulates the transcription of certain segments of DNA into mRNA. The mRNA leaves the nucleus through nuclear pores and in the cytoplasm is translated on ribosomes into proteins that allow for more sodium reabsorption and potassium and hydrogen secretion. In particular, it increases the formation of sodium-potassium pumps on the basolateral membrane of principal cells. It also increases the expression of epithelial sodium channels, or ENaC, on the apical membrane of principal cells. The action of ENaC and sodium-potassium pumps allow for movement of sodium from the filtrate back into the renal capillaries, so less sodium is lost in the urine. Sodium attracts water. Therefore, the end effect of aldosterone is to increase serum levels of sodium and to increase the fluid volume in the vasculature to increase blood pressure.
Okay. Hang on. I don't know how to get out of this and get back to the presentation. Hang on. There we go. All right. Anything else that you have to share with us about this one? Nope. All right. The next hormone we have in this sequence. Let's see. Come on.
As aldosterone is
No,
hydrophobic, it diffuses.
All right. Here we go. All right. So next, epinephrine.
Okay, so source of signal, commonly referred to as adrenaline, secreted by the adrenal glands and medulla in the brain. And then the type of signaling, the endocrine system, because it interacts with the sympathetic nervous system. The type of receptor, epinephrine binds to a G protein coupled receptor. And then could you play the video?
Yep.
The following video describes an example of a G protein receptor cell signaling pathway. Epinephrine is a hormone commonly associated with the fight or flight response. When an organism is faced with stressful conditions, the brain releases the hormone epinephrine. epinephrine travels to the liver cell to cause a response. Because epinephrine is a ligand, it can't go through the phospholipid bilayer membrane, so the epinephrine binds to the G protein coupled receptor, which marks the first phase of cell signaling, reception. The binding of epinephrine causes the G protein coupled receptor to change shape. This allows an inactive G protein to bind to it. This is the beginning of the transduction pathway. Then, GTP replaces the GDP on the G protein. The activated G protein binds to adenylyl cyclase. The job of adenylyl cyclase is to convert ATP to cyclic AMP, or adenosine monophosphate. Cyclic AMP serves as a second messenger by helping to activate protein kinases. Protein kinases have two parts, the regulatory protein and the catalytic protein. The regulatory protein stops the catalytic protein from working. The cyclic AMP binds to this regulatory part to separate it from the catalytic protein. ATP then donates some phosphate groups to the catalytic protein in order to activate it, in the process called phosphorylation. This catalytic protein goes on to donate those phosphates to phosphorylase. The function of phosphorylase is to break down glycogen into glucose molecules. This marks the end of the signal transduction pathway. The final phase, response, is when the glucose travels out of the cell through glucose transporters straddling the membrane.
All right. Go ahead.
So the epinephrine will bind to a G protein on the cell's surface, which causes the receptor to change its shape and convert to its active form. And the G protein will then bind to another receptor.
All right. Anything else? Okay. Then the next one we have here.
The following video describes an example.
Sorry. Having technical difficulties today. All right. Human growth hormone.
Okay, so can you guys hear me okay?
Yep.
Okay, so the human growth hormone is a key growth signal released from the pituitary, which is a pea-sized gland located at the base of the brain. And it's an endocrine signaling because it happens when the hormones are produced slower and they last longer. And then it's a cytokine receptor because it lacks natural protein tyrosine kinase activity, and therefore it relies on binding to non-receptor BTKs. And then,
So the cellular response is after the hormone is released from the pituitary gland. Another hormone is created in the liver, and it's like similar to insulin in a way. And this stimulates the production of cartilage cells, which eventually leads to like bone growth. And another info about it is it used to be harvested from cadavers, but a lot of the scientists that were harvesting it got a disease called Creutzfeldt-Jacobs disease, and so now we use a synthetic version of the hormone.
That's really interesting. Do you guys remember Creutzfeldt-Jakob is like the mad cow for humans? It's that same kind of thing. I never heard that before. That's really interesting.
And then we also have a little video.
Okay. Does it have sound?
Yep.
Okay. I think it'll, yeah.
Today we'll be explaining the cell signaling pathway for the growth hormone, which causes bone and muscle growth as well as stimulates cell reproduction and cell regeneration. The growth hormone cell signaling pathway takes place at the pituitary gland, where it connects to the tyrosine kinase receptor by binding at the signal binding site. Once it has binded at the signal binding site, the two signal molecules combine together to create a dimer protein, which causes the tyrosine kinase to change shape and activate. Dimerization activates JAK2, which stands for Janus family tyrosine kinase 2. The main function of this enzyme is to transfer the phosphate groups from the ATP to the protein within the cell. So the ATP contains the ADP and these phosphate groups, and these phosphate groups are then connected to the JAK2 tyrosine kinase, which causes the whole receptor to become fully activated. Now that the receptor is fully activated, the STAT proteins floating in the cytoplasm attach to the remaining phosphate groups. Then JAK2 phosphorylates the STAT proteins through ATP once again, and the two STAT proteins then combine together to create a dimer protein. This STAT dimer enters the nucleus and binds to the DNA, resulting in transcription and cellular responses as well as gene expression. Within the body, the growth hormone directly affects the adipose cells, which break down and release fatty acids and are metabolized and cause an increase in blood glucose, which stimulates cell growth. The indirect effect takes place within the liver, where the growth hormones travel through the blood and stimulate the liver to produce the insulin growth factor known as IGF-1. IGF-1 causes growth within bones and muscles in the body.
All right. Any more to add there, ladies? Akila or Alyssa, anything else?
Nope.
All right. Then moving on here. I don't know why this always does this. Okay. And then insulin, Ryan and Lincoln.
Yep. So we had insulin, and insulin's a signal produced in the pancreas that regulates blood sugar. It's a type of endocrine signaling because it goes through the bloodstream and acts on distant cells. And the type of receptor for insulin is a ligand-gated ion channel, but it's also a type of tyrosine kinase, because the way it was explained to me is that as the ion channels open, like I guess phosphorus goes through or phosphate goes through at the same time. So that's kind of how it's both. And Lincoln has the other half.
Right. The cellular response is the secretion of insulin from the pancreas directs the muscle and fat cells to take in glucose. The cell then obtains energy from the glucose. And some other info, if insulin is depleted, the body cannot regulate the amount of glucose in the blood.
And we have a little short video. It does have volume.
Okay. It seems to be working okay now, so. Usually, hang on. That one played differently, so I got to do something different. There we go.
When food is ingested, it travels along the digestive tract, where it is broken down into its component nutrients in order to be absorbed into the bloodstream. One such nutrient is glucose, a simple sugar. Glucose gets absorbed by the stomach and intestines and then enters the bloodstream. It travels through the circulation to all body cells. Once absorbed into the bloodstream, glucose circulates, causing the blood sugar level to rise. An increased level of blood sugar sends a signal to the pancreatic beta cells, which respond by secreting the hormone insulin into the circulation. Insulin is necessary for glucose to reach and be used by several important target tissues throughout the body. These include the liver, muscle, and adipose tissue. Insulin is necessary to keep blood glucose levels stable in the body. Glucose enters the cell through a process called facilitated diffusion.
All right. Anything else, Lincoln or Ryan?
I don't think so.
All right. So I believe that was all of the different types of hormones. Obviously, there are lots more. These are just a few examples of each of these. The last kind of group of signaling molecules that we are going to look at are the plant hormones. What just happened? Where did my slide go? Oh, here we go. And plant hormones are a little different. They don't totally match the definition of normal hormones, but they're still chemical signals that can modify or control physiological processes in plants. And so we're going to take a look at just a couple examples of these. And then we will go back to that one that we skipped a little bit ago if we need to do that. So let me get back to the right place in our presentation here, since I kind of did these out of order. Give me just 1 second, and we will go back and talk about 2 common plant hormones. All right, so Brianna and Abby.
Okay, so auxin is synthesized in the plant cells of the bud, the stem, and the roots, and it actually moves to the side of the plant that is darker and causes those cells to grow. So the whole plant leans towards the light, which is phototropism. And it's a phytohormone, which is just another way of saying plant hormone. And it doesn't really match up with one specific type of signaling, since it can be excreted to other cells, but also used in the cell that it was produced. And it's received by the F-box protein, TIR1, both inside and outside of the cell.
And then kind of the pathway that it goes through, it first goes to like a receptor in the plasma membrane, and then it like stimulates growth in that area. So it'll like mess with the plasma membrane's proton pumps and then bring like a whole bunch of water into the pathway, into the cell, and that'll cause the cell to grow. And then again, like I said, auxin activates the proton pumps, and that changes the pH of the cell and allows the enzymes to weaken the cell wall. This process calls the degraduation of a growth depressor and results in the growth of a cell. It's basically what I just said. And the hyperpolarizations, the members, that makes it like more, I think it makes it more negative, and then that causes that water, that osmosis to happen, and that'll make the cell grow. And again, this is auxin, so it has like lots of different features. It's just a general growth hormone. And auxin can regulate gene expression. So that's kind of, there was an experiment done where you like cut the tip of a like growing plant off, and then you see which way it grows, like based off of like the auxin inside of it. And also auxin is used as, another fun fact, auxins can be used as a herbicide if you spray it on certain plants like weeds. And then we have a little video link, and I think we only need to go to like the 1 minute mark, if I remember correctly.
All right, let me swap this over here.
Plant hormones, also known as plant growth factors, are chemical substances that accelerate, inhibit, or otherwise affect growth. This animation explores the action of indole-3-acetic acid, IAA, the most common form of auxin, and looks at how we discovered what auxins do. The auxin IAA is produced at the apex of the stem. Under unilateral light shining on one side, IAA is redistributed to the shaded side, creating a gradient of the hormone across the stem. IAA travels between cells by diffusion. IAA activates proton pumps, which lowers the pH. This activates enzymes, expansins, break bonds in the cell wall. The loosening of the cell wall allows water to enter the cell and turgor pressure increases. Cells on the shaded side become elongated and cause the shoot tips to bend towards the light. But what is the experimental evidence for this? Darwin investigated using oat coleoptiles. He found that there is no bending response to light when the coleoptile tip is removed.
Do you want me to stop there? Or do you want to watch more?
You can
That's
pause it.
Okay.
Yeah.
All right. Anything else on here?
Nope.
All right. Then the other plant hormone we have, Annie and Chloe.
Okay, so the source of signal comes from the roots, like young leaves and seeds, and then the type of signaling is like the endocrine signaling.
And then the type of receptor is a GID1 receptor, and there are two types, soluble and membrane bound. In the cellular response, it is used to stimulate growth and development of like organs, so like the stem and stuff, through enhanced cell division and cell elongation. So basically just plant reproduction, like the cell reproduction in the plant. And just a little fun fact of it, GA is like the name for the hormone that I can't pronounce. And the role of it determining plant stature had a major impact in agriculture in the 1960s. So they like used it to help agriculture.
Awesome. Anything else? All right. Is Macy here or not?
I don't think so.
Emma, would you like to just give us a brief synopsis so that your part of the presentation is done and then we can have her tell us a little more later? Or do you want to wait and do it Monday?
I'd rather wait and do it till Monday because most of the important things are, I just have the summary of the pathway, so.
Okay. I will get in touch with her then and we will do that one on Monday. All right. So just one more quick thing to kind of wrap up here. Let me switch my presentation over a little bit here. So they talked a little bit about that and we talked about those. There's one more example that's linked in the slides here. If you ever want to go and watch this, it's kind of interesting. You talked about many, many, many examples of the cell signaling within eukaryotes, within animals and plants. Prokaryotes, bacteria also do this type of signaling amongst themselves. And obviously there it's like communicating between different organisms since they're single-celled creatures instead of within one multicellular creature. So that TED Talk is linked to this picture. If you go into the slides and click on that, I think this one's about 15 minutes. That's about what most TED Talks are, but it's pretty cool. So the last thing for today, there is a brief reflection that I'd like you to do. It should only take you 5 to 10 minutes, especially while it's all fresh in your brain if you want to do it right now. It's a Google form. I linked it on today's lesson post. I didn't even think about putting it separately on the homework. So it's in today's lesson. If you have a couple minutes right now before 5th hour to go in and do that, that's kind of the wrap up for this class activity today. So feel free to stick around.